So with terms of the Reflex Lab, we have done part one. Part one, which was, hopefully that's all in your lab notebook. You've got some conclusions, some discussion written up about that at this point. The next part two, are just demonstrations. Normally you would do these with each other. Instead, we're just gonna kind of go through them through like videos and stuff. I do recommend trying them at home if you have the ability. We'll talk about that a little more as we do this stuff. In fact, one of the ones we actually can do all together when we get to the balance thing. But we will do that all together in a little bit. But we're going to start with ones that you're just going to see. So I'm going to go and share my screen. Here we go. All right, so doing the Reflex Lab. So this is all stuff that's just in that module. Again, oh, and I still gotta update this Excel spreadsheet so it doesn't say I forget what is it, seconds or something. I gotta fix that. So part two, demonstrations of reflex. So we did the plantar reflex and we did the corneal reflex last time. So those should be done. Next, we're going to get to the pupillary light reflex. So this it was a little frustrating for me. I looked on all sorts of YouTube videos trying to see good images of people doing this reflex where you could clearly see what was going on in ways that you needed to see it. And the lighting is so tricky because it's, when you are shining a light, then the other eye gets like really dark looking. And so I could not find any of the videos with real subjects where you could really clearly see what you needed to see. So this thing that I'm going to play here is a simulation, but it's what you would see. Like, the fact is your real visual system is really good at dealing with different brightnesses and contrasts. Like, imagine, I don't know if most people have had the experience of trying to take a picture of some scene and you're looking around and you can see it. Oh, there's the sun, there's this bright, that's in the shade, la, la, la. But you can see it all. You can make out, what's going on. And then you try to take a picture of it and you're screwed because it's like, either this is I can see this, but this is like black, or this is now visible, but now that's so washed out, it's white, right? So it's your visual system is way better at dealing with things that are on this huge range of brightnesses than a camera is. And that's kind of what we run into the problem with trying to make a video of this, is that you can, at least I think you could probably make it work, but maybe that's something I'll do if I ever get extra time. I'll try to make a video on YouTube people can use. Before we get into the details of pupillary reflexes, let's just review just for a moment. Here's the eye. Oh, hold on. Okay, let me mute you. So the eye, and This is the iris. The iris is, again, most of you have been in anatomy. It's got smooth muscle, the involuntary muscle. It's got some fibers that are going longitudinally. And when those are going to constrict, it's going to, dilate the pupil. Now that we have brought in all these divisions of the nervous system, which division of the nervous system is responsible for dilation of the pupils?
Sympathetic.
Parasympathetic.
Again, the way I usually remember it is like, again, kind of wide-eyed with fright. Like, when somebody is like, whoa, like their eyes are gonna be like, get all big, right? So that fight or flight is dilating your pupils. For pupillary constriction, that's gonna be parasympathetic. Some of the questions for these little like mini discussions for the different things, ask you specifically for some of these reflexes, which division is mediating this response. So you need to make sure that you kind of remember this. For constriction, there are these circular fibers that will constrict, which is going to make the pupil, the little hole, smaller. And this, in general, it's constantly, I didn't mention it a few moments ago, but I'll mention it now. Those sympathetic and parasympathetic, it's not like one is on or the other is on. Usually it's, people talk about it's like driving with one foot on the brake, one foot on the gas, and you're kind of adjusting them. As the light intensity in the room changes, if it's getting a little dimmer, your eyes will dilate to let some more light in. If it gets a little brighter, they'll constrict to block some of the light. So it's not like your eyes are either dilated or constricted. They're constantly kind of adjusting to whatever they need to do in order to, have the right amount of light coming in. That being said, part of the function of this is it's like the iris on your camera. It's adjusting the amount of light that's coming in. If it's super bright outside, what's the pupil going to do?
Constrict.
Constrict. And as it gets dimmer, it'll dilate. When you're doing these reflexes, you want to start with the room not that bright, because otherwise the pupil is totally constricted, so you can't really see it getting closed, getting more or less open or closed. Again, which is part of the reason these videos all kind of stink, is because they're doing it under low light and then shining a flashlight in somebody's eye and the camera gets all messed up. So what would you expect if you shine a bright pen light in somebody's eye? What are you going to expect the response going to be?
Constriction.
It's going to be constricting. Then we use those words. If the constriction only happened on that one side, what would we call that?
Ipsilateral.
Ipsilateral response. When you do this, if you're doing this at home with somebody you're sheltering in place with, it's fun to do. You're going to take like a pen light, which you can then shine in their eye, but you want to block the two sides of, let me stop this one now. You're going to block so the two eyes are not getting the same stimulus. And you can then just use like a millimeter ruler, and you can try to figure out about how many millimeters is their right pupil, how many millimeters is their left pupil. And then this is actually easier if you have yet another person, a third person, come in and shine a light in one of their eyes. So then as that light is in the eye, This is hard for me to do. You want to now measure what is the size of the pupil on the side that the light's shining in, but also on the side that's not having the light. Right? So does that make sense? The idea here is you're shining the light, only one eye is getting the stimulus, but you are looking for a response or no response in both eyes. And you need a pretty bright light to make this work. But if you have never actually looked at this, you'll see this in the little video here, but it's really trippy to actually see the consensual reflex if you've never really thought about it, because it's not that intuitive at first. It is once you think about the wiring and all that. One thing to think about when you think about kind of the wiring of the eyes to the brain. So let's say this is your brain here. This is your nose. These are your eyes. The way the eyes are connected to the brain is each hemisphere of the brain is responsible for 1/2 of your visual field. Again, we're going to talk about this in more detail, but basically you have kind of the right visual field over here. You have the left visual field over here. And the eyeball gets information from this part of the visual field. This eyeball also is getting information from this visual field. And both of those eyeballs go into the same side of the brain. Meanwhile, each eyeball is getting information about the other visual field. And those are going to go into the other side of the brain. So each side of the brain is getting 2 versions of each hemifield, each half of the visual field. So like the left side of the brain is actually getting information about the right hemifield, but from the left eye and from the right eye. The right side of the brain is getting information about the left hemifield, but from the right eye and the left eye. If you all remember the optic chiasma from anatomy, it's where these neurons kind of cross over and make that X right underneath the pituitary gland there. So what is this kind of telling you though? If you shine a light into one eye, does the stimulus just stay on one side of the brain, one side of the body? No. Yeah, exactly. If I shine a light into one eye, it's going both sides of the brain. So it's going to make sense that the response is going to come back on both sides of the body, right? So the stimulus in one eye goes to both sides because each eye is actually sending information to the 2 hemispheres about the 2 hemifields, or half a visual field. So just to kind of make, kind of motivate why it's going to make sense that the response is not necessarily going to be confined to the eye that's getting the light. So here we go. I'm going to share this thing and we'll watch this little deal.
Hi, a normal pupil examination can be documented as being Pearl and no RAPD. This shorthand states that the pupils are equal and reactive to light and that there is no relative afferent pupil defect. So how do I actually test these properly? I ask the patient to fix in the distance, then I check the pupils are equal in size, and again with the lights off. Then, with the lights still off, I check each eye has a direct response to light. Fine.
So do you see, I mean, this is happening kind of fast. That arrow is supposed to be like the flashlight. I mean, I'll go back there. Watch what happens to the eyes. Whichever one is getting the arrow, that's where the light is going. But what you're seeing is the pupils responding to the light going in.
Then, with the light still off, I check each eye has a direct response to light. Finally,
So do you see that? So that's what actually happens. Again, it's hard to see that on the videos of real people because of the contrast. That's why I have this silly cartoon thing here for you. But I want you to really get a sense that both pupils are responding dramatically to the same stimulus. Doesn't matter if it's the eye getting the light in it or it's the eye on the other side. So now we can continue.
I do a swinging light test to check there is no RAPD. Now, let's recap with a little more explanation. Firstly, to avoid the near reflex where the eyes converge, accommodate, and the pupils constrict, ask the patient to fix on an object in the distance. You should check the pupils are equal in both light and dark, or you may miss an abnormally small pupil, such as is seen in Horner syndrome. For more on unequal pupils, watch the video titled Anisocoria. When examining the pupil reactions, having the patient in the dark with distance fixation makes the pupils as large as possible and makes the pupil reactions easier to see. When you shine the light at the pupil, watch the same pupil for a quick constriction followed by a slight relaxation. There is no need to look for a consensual reaction here, as both eyes have demonstrated a direct reaction indicating an intact afferent pathway from eye to brain and efferent pathway from brain to pupil. There is also no need to check for a response to accommodation unless the pupils fail to respond to light. Finally, the swinging light test to check for relative afferent pupil defect, or RAPD. This is a comparative test of the two optic nerves and may detect conditions such as optic neuritis or optic nerve compression, where the nerve is functioning but poorly when compared to the other side. Again, this should be in the dark with distance fixation. First, shine the light at the first eye. The pupil will constrict and then relax a little. Now swing the light source quickly and directly to the other eye. The pupil will have just started to dilate when the light hits it, causing a small constriction, followed by a relaxation. Make sure you hold the light on each eye for 2 to 3 seconds to allow for each pupil to first constrict, then relax before you swing the light to the other eye. Remember, your light source must be really bright to reliably detect an RAPD. A standard direct ophthalmoscope or pen torch may not be bright enough. Okay, you've now examined these normal pupils. To criticize...
So don't worry so much about the RAPD, but the main thing here is to, and again, and in your lab write-up, you're not going to have actual like millimeters, but you should talk about what you would expect in a normal response. And this is also where I don't get the opportunity to, hold on, let me, there, stop sharing. I mean, I have, in fact, last semester even, it was ridiculous. This is another one of these things about, that kind of sanity check. Like if you read in their lab book, it talks about like, not having, these, basically if it's not having either the corneal reflex or this pupillary reflex, they're like kind of ominous signs. There's something really wrong. So if you are doing this on somebody and it's like, oh, it's not working, it could be you're doing something wrong, but you don't want to just say like, okay, let's write down it doesn't work and move on, which I have had students do in the past. It's like, okay, so you are, either you're doing something wrong, in which case we should look at this, or we need to get your subject to the ER because something's really wrong with them. So if you're doing this at home, you should see these. These ones aren't tricky to make happen. If you are in kind of dim light and you shine a bright light in their pupil, it's going to constrict. You will see a consensual response. As opposed to like when I was saying like the plantar reflex, that one's a little trickier, but this one's not tricky. So you should definitely do that. The other one was, the next one actually is pretty tricky, the ciliospinal reflex. General, let's go back. So this one is elicited by pinching, usually the back of their neck. And the response, what do you think? What do you think is, what kind of response will you probably get to, like, oh, getting a pinch? Is that going to be sympathetic or parasympathetic?
Sympathetic. Sympathetic.
That's kind of more fight or flight. It's like, ouch. And what are you going to expect the response going to be in the pupil?
Dilate.
Dilation.
Dilate.
So I will play this for you. I will warn you, in reality, seeing it happen in this video is kind of like, I can pretend to kind of sort of see it. And even doing it in real life, like, it's maybe if you're clinically trained more, it's easier to really notice it. This one I find way less obvious than the pupillary reflex that we just talked about. But I will show you this because it is an important clinical test.
Hello and welcome to Online Anatomy. So today we're going to be talking about the ciliospinal reflex. In order to elicit this reflex, you can do this with yourself or with a partner. If you're doing this on a partner, you're going to look into your partner's eyes, and then you are going to stimulate one side of the back of the neck, right, by gently caressing. So this is a wonderful bonding experience. If you're doing this on yourself, you can pick one side of your neck to stimulate while looking at yourself.
It's typical. I mean, she's talking about this gentle caressing thing. Maybe it's most of the time you hear about it being pinched or being kind of scraped by something kind of sharp. So, I would say if it works from a gentle caress, that sounds way better. But often when I hear about this one, it's not quite as kind of sweet as she's making it sound here.
Or recording yourself. So I'm going to stimulate the left side of my neck, and I want you to pay attention to what is happening in the ipsilateral, so my left eye, the same side that I'm stimulating, and the contralateral, the opposite eye, and what's happening to my pupils. Okay, so I'm going to go ahead and turn this bright light off and get uncomfortably close to the camera. All right, so I'm going to just gently rub the back of my neck and see if we can get a response. Now, if you need a little bit more stimulus.
Right, so it's like, you can't really see it. You can't even see what's going on in her other eye. So, but pretend.
You can pinch the back of your neck, and we'll see. So what you're looking for is the ipsilateral pupil. You're looking for the ipsilateral pupil to dilate, to get bigger, right? So this is different than what we saw with the pupillary light reflex, with the contralateral pupillary light reflex, right? With the pupillary light reflex, if you shone a light into the one eye, you're not only going to get a response in the ipsilateral eye, but also in the contralateral eye. So if you block off or divide the eyes, so that way the light that you're shining light into one eye, light is not coming into the other eye, you're still going to elicit a constriction of the pupils in the contralateral eye. This is because constriction of the pupils is parasympathetic. It is a parasympathetic innervation response. So the parasympathetic innervation of the pupillary muscles are closely integrated. With the ciliospinal reflex, this is dilation. So dilation is a sympathetic nervous system response. So it's sympathetic innervation, so on the ipsilateral side. So that tells us that there is not very close integration between the two irises with sympathetic nervous system integration.
Okay. All right. Da, 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 da.
I'm asking you to make a donation right now to have your gift.
Shush. So, yeah, I mean, it's hard to really see it there. You can, I'd recommend look in the mirror and you can do a pinch. You could try a caress, try more of a pinch, more likely. Even then, it's subtle. Don't feel like a failure if you're having trouble really seeing the ciliospinal reflex. I would ask you, since this is something you can do on yourself easily, do it. Go look in the mirror. Everybody's got a mirror. So describe what you see if you see it. Describe if you don't see it. That's all right too. You can say, I tried it, but it's kind of subtle. I'm not noticing it. But then you do need to write in your lab notebook, what is a expected response and why. So this is one you can do. I would like you to actually write this in your lab notebook, what you see when you look at yourself in the mirror as you kind of pinch or gently caress the back of your neck. Usually they say like near the hairline. So the next two ones are about equilibrium. And so we need to talk a little bit about the inner ear. So when we talk about equilibrium or about balance, how do I know, how do I not fall over? There's a few things going on. We're going to look at the ear in much more detail when we are getting into the sensory part of the nervous system. But for right now, we need to introduce the saccule and the semicircular canals. Probably easiest, I can just, let me find some videos here. I'm going to go share screen. So this is the, in fact, I should probably show this, I should show this kind of in the head first. So this is like your ear. So this is your head. The inner ear is here kind of within your skull. So whenever you are moving your head around, you're moving the inner ear, which is embedded in your skull. So it's important to remember all the stuff we're talking about. If my head is moving, my inner ear is moving, because the inner ear is embedded in my skull bones, in the temporal bone of my skull. Within the inner ear, there are what we call the vestibular organs, and there are two different things that help you with your sense of balance. One is the semicircular canal. So let's look at those first. Wow. Wait, let me find a better one. So these semicircular canals, you have 3 of them. And again, they're embedded in your skull. So whenever you're moving your head, these canals are moving along with your skull. And each of these canals, there's 3 of them. They're all exactly the same except for their orientation. You can see they're like in the 3 different axes. They are going to be responding to rotational movement. So is my head moving what we call like, yaw or pitch back and forth or roll back and forth? So you have 3 different axes. Each of these semicircular canals is going to respond to your head rolling or pitching or yawing. So each semicircular canal it's got this fluid in it called endolymph. And this fluid has inertia. So remember, everything out here is my skull. So if I am moving my head around, I am moving the skull, but the fluid has inertia, right? Inertia, if let me, here is a glass of water and it has water in it. If I start moving the glass back and forth, does the water actually spin with the glass?
No.
The water kind of stays still. It has inertia and the glass is moving, but the water is actually not moving with the glass. If I keep moving the glass around and around and around, eventually the water is going to get entrained with the glass and start moving with it and start kind of getting a little bit of a current. But same thing in your skull here, when you move your skull back and forth, the skull moves, but the water, this fluid, stays still. It has inertia. And you have this little gelatinous membrane called the cupula, which again, we're going to look at in more detail. For right now, don't worry about the name. You've got these special little mechanoreceptors, little hair cells that determine bending. So these basically have an electrical signal if you bend. So this thing here, this is going to detect bending. So if I am, and this is moving, so if I'm moving my whole head, let's pick one direction to make this more obvious. So let's say I move my head this way. So this is moving this way. But the water is not moving. The water is staying still. All of this water here is not moving. So that means that this little cupula is going to end up getting pushed the opposite direction. Here, let me delete the background just to make this easier to show. Like here, if I have, this is water. This is water in a bowl. Here I have some floppy membrane and it's sitting in the water. If I move this direction, the water's not moving, it bends back the opposite. If I move this way, So this is kind of what's happening. The cupula would be like my orange paper here, and it's attached to the whole rest of your semicircular canal, and you move your head so everything moves, but the water doesn't. This little cupula, this little gelatinous curtain, bends the opposite direction of whichever direction you are actually rotating your head. And that is going to send a message to your brain that, you know, your head is turning. So if I turn my head this way, the little cupula is going to bend the opposite direction, and I get a message that my head turned. If I go this way, I get a message that the head turned the other way. I wonder if I could do this, actually.
Could you share that again? Because you're still sharing that diagram on the screen.
Oh, shoot, I'm sorry.
If you double click on his box that he's shown in, it'll full screen him.
All right. I should be full screen for everybody now? Okay. So the idea, you've got the water that doesn't move. You've got this kind of floppy membrane which is attached to the rest of the skull. And then my whole skull moves in one direction or the other direction, but the membrane hits the immovable water and bends in the opposite direction. That bending is detected by those little hair cells which create an electrical signal to the brain, tells the brain that, oh, the little thing is bending, which means my head must be spinning. Again, we're going to look at this in more detail, but we need to kind of introduce the idea right now. And again, you have these little semicircular canals in all three axes. So I can tell if my head is spinning this way. I can tell if my head is spinning this way. I can tell if my head is spinning this way. Rotating, I should say. What happens, I can just do, what happens if I, make sure I'm not hurting myself. If I stand up and just start turning and turning and turning. Actually, I should show you, before we do this, let's talk about the reflex. So one of the reflexes that's connected to this is just maintaining gaze. So let's say I want to keep looking straight ahead. I don't, can you see my eyes right now?
Yes,
Yeah?
we can see them.
So let's say I want to keep staring straight ahead, but I start moving my head to the right. My eyes have to roll to the left to keep looking there, right? Which if I keep rotating my head, if I want to keep staring straight ahead, I need to keep rotating my eyes within my skull to maintain my, if I'm going down or up. Whichever way my head rotates around, the eyes have to compensate so I don't lose what I'm staring at. Do y'all see that? So that is going to be important because, you know, if I'm not moving at all, my eyes don't move in my head. But if my semicircular canals are fooled for some reason and think I'm spinning even though I'm not, my eyes are going to start doing weird stuff. And that's actually another important reflex that you need to know about. We're going to call nystagmus, is when the eyes start flicking around weird because your semicircular canals are getting information that you're spinning even if you're not spinning. So like I think I can do this. You know, I'll spin. You know, if I start spinning, eventually as I keep spinning here, what is happening to the fluid inside my semicircular canals?
It will start to move
It's going to
in your direction.
Exactly. So at this point, I don't feel dizzy at all because the fluid and my skull are actually moving together. So there's no bending of the hair cells. So I don't feel dizzy. What happens if I stop all of a sudden?
You're going to feel super dizzy
And it fall down.
Why is that going to happen though?
because the liquid inside the canals, it has some movement.
And it
And
stops all of a sudden.
Yeah,
it's gonna crash with the hair
exactly. The
cells.
moving and hit the hair cells, and my brain will think I'm spinning even though I'm not, and my eyes will start. So I'm gonna try to do this. I'm gonna look straight into the camera right now.
Oh my goodness, sorry.
Wow.
Can you see my eyes?
They were doing like this.
Yeah, so that's nystagmus, that flicking of the eyes. That is something that you can make it happen different ways. You can do caloric nystagmus. You can pour warm water in somebody's ear, and that sets up convection currents in the fluids, which will cause the eyes to start flicking. But that's another, and that's something you can do at home with somebody that is game. Have them spin around and then have them stop and look at their eyes and watch what happens to their eyes. There are other things that we use to maintain balance. So we know whether our head is spinning. There are also the sacculus and the utricle, which we'll talk about in more detail later, which tell you which way gravity is going. Your muscles tell you kind of where, which is happening. Remember we talked about the stretch detectors. Your muscles will tell you as your body moving over. What else do you use to make sure you you don't fall over?
Sorry, Professor, what was the question about the muscle?
What are the different senses you use to kind of keep your balance?
Your eyes.
But one of your eyes as well. Just, looking around, you can kind of tell like, whoa, I'm moving. I better kind of pull back in.
Also, sense of touch. Do you think sense of touch would be it? Because you kind of, when you're dizzy, you kind of touch around to like look touch what's around you.
I'm thinking more of not doing that. So I think that there's things in the inner ear that we're going to look at in more detail. Just the sense of your muscles, whether they move or not. And then your eyes as well. It's like, so part of the lab, which I am actually we do have time for this. So what I'm gonna do, if you look at 2E, which is equilibrium and balance reflexes in there. What I would like you to do, you're going to be able to see each other in Zoom. I'm going to just throw everybody into some random Zoom meetings right now. And I want you to just, pick somebody in your group to be the subject. And it's basically you have them stand with their feet and arms together, arms outstretched. And this is not having them spin. Don't have them spin. Have the subject stand with eyes open and eyes closed. And again, observe, record body sway and corrective motions required to maintain balance. So you're going to just write down in your lab notebook, what do you see with their eyes open? Maybe they're just like rock solid. Their eyes closed. Have them stand on one foot, again, with eyes open and eyes closed. And this should only take like 5 minutes. So I want you to actually do this. Again, I will, I'm gonna make the breakout rooms. We're gonna, were people a little surprised at what they found?
No. Not really, because if you eliminate all these senses like your eyes and stuff, then you don't have as much information telling you about your like body perception or proprioception.
Okay, yeah, I mean, because you do have proprioception, there's a lot of sense of what your body's doing, and you have your vestibular organs, which are telling you also which way is gravity, but in general, they tend to be kind of slower circuits, as opposed to the visual ones, which are much faster. I mean, I know the first time I did this, I was shocked at how much worse I was keeping my balance with my eyes closed compared to keeping them open.
I couldn't, mostly. My eyes closed, I was moving like a bimbo.
Yeah, so I mean, I mean, definitely, I know at least for me, it was kind of, it's kind of a bad pun, eye-opening to see how much my sense of balance actually was relying on my vision to actually feel solid. You know, if you do yoga poses or something where you're balanced, they always tell you to kind of fixate on something, you know, and it kind of makes more sense now where you kind of realize how much your vision is really tied into
100%.
yourself upright.
And the same thing with dancers, actually, when they're turning.
So the turning is also, that's a little different. The like spotting, that is so like you don't get dizzy, right? You know, when
Nice.
right, remember how I was spinning and I'm spinning and spinning and spinning, and then I stopped and my eyes are going, and actually I was holding onto my desk so I didn't face plant. But if you are in some dance and you want to be spinning, you want to come out of your spinning and then just go into your next like move. So there, what you're doing is doing the spotting, you're trying to keep the fluid from actually starting to match your skull. So you're turning, but not moving your head. And then you flip your head super fast. And then you turn your body, but keep your head still and move your head really fast. You know, and by the time you're done with all that, and then you stop, the fluid has never gotten sped up with your head. So you're ready to just do your next move. Right? So that spotting is about keeping the fluid from catching up with your skull by doing that spotting and just flicking your head really fast. And it's kind of like the inertia, like poof, the fluid didn't speed up, the fluid didn't catch up, the fluid didn't catch up. Whereas if you're just constantly slowly moving it around and around, eventually the fluid's moving with your skull. So that's like that spotting. So, you know, gag reflex, you know, observation of nystagmus. Let me go share. So you've seen nystagmus. There's also a little video. There's kind of this goofy guy and a quick little video that you can see doing nystagmus. You definitely, like in your lab notebook, you can describe what you saw with me, or go to that little video. It says goofy nystagmus video. It's silly, but it works fine. Gag reflex is another thing that, it's probably like in college is where you like kind of learn gag reflex. Just like, oh, I'm so sick. People like put their finger down their throat and vomit, whatever. That's, kind of the gag reflex. If you kind of stroke the upper part of your palate, the whole thing kind of pulls up. And it's actually a clinical test to make sure like your cranial nerves are functioning properly. And you can just use like a little, like a tongue depressor or something, and you can just see the whole, in fact, what time is it? Is enough time. I'm going to share my screen here.
Nystagmus demonstration. We have Brian Shealy. He's going to be spinning to the left.
Oh, this is that goofy spinning guy. So you can see he's got like nystagmus.
Hey, now. Oh man, it's good.
So you have somebody doing nystagmus as one of the little videos. Here's the gag reflex.
The reflex action, we're going to actually look at the gag reflex. Have you open your mouth, Tilt your head back just a little bit. Good. And it's going to gag you. That is a good gag. Okay. The reflex action.
So, there you saw a gag reflex. Again, you should just describe it. Describe it, because part of what the point of doing this is just to get a sense of what are some of these innate reflexes that are used as part of a neurological exam, where you know what the correct response should be, and if it's not there, then there's information that there's something wrong with your nervous system. So I think that we're going to leave that. There's something in your lab manual about conditioned reflexes. We're given the time. Let's not look at that. The basic idea there is everything we've talked about so far are innate reflexes. There's a lot of reflexes that are just automatic. You shine a light in your eye, your pupil constricts, and you don't have to learn it. It's just something you're born with. There are other reflexes that are not things you're born with. Your nervous system makes the connection. You can do things where you shine a light in your eye, the pupil constricts. They call that the unconditioned reflex. I could then have something where I'm, like some bell that I ring. And I ring a bell, and that's not usually going to make my eye constrict. But if I pair them together, we call this, so if this happens enough, where you have some stimulus that normally would not cause the eye to constrict, but it's always happening at the same time as the light goes on, eventually my nervous system actually starts rewiring to have my eye constrict just from hearing the bell. That's that famous Pavlov's dog, right? Pavlov's dog is a conditioned reflex. The dog smells meat and salivates, and normally a bell doesn't make a dog salivate, but eventually if you have the meat powder and the bell and the meat powder and the bell and the meat powder and the bell, eventually you just ring the bell and the dog starts salivating. So reflexes can also be conditioned as well, but you don't have to have that as part of your write-up. So that's just, all right, so in the last few minutes, let's talk about the nerve stimulation lab. And there is stuff on, well, I'm going to go into share screen. Let's get out of Goofy Nystagmus guy. And I do have, I created a submit reflex lab write-up. That's due like in 2 weeks. So it's like 2 weeks, you should have that turned in. So for nerve stimulation lab, there's an introductory thing that you should watch. We'll give more detail. I'm going to talk really quickly, but there's more information here, so it's not like there's nothing else for you to look at. I do want to bring in a couple of points just to give you a little more information to put together with that video. So that's what I'm going to do in the next 2 minutes right now. So the basic idea, here's like your brain and spinal cord. Okay, whatever. Let's say here's your finger. And then you have these specialized little receptors that are in your finger. If you've been in anatomy, you remember like the little Meissner corpuscles and things like that, the little touch receptors. And then you have a axon that carries a message and eventually goes up into your brain, into your parietal cortex, where the signal comes in saying, oh, I'm feeling touch from that receptor. And normally the way it works is, you get a touch, and the touch is what operates or activates the action potential, which goes up and gets to your brain. But you can also just put some current source here. So use some electrodes, put some electric current across, which is going to create an electric current through your tissue. As long as there's enough to bring this neuron above its threshold, that is going to activate an action potential, which will go up to your brain and send a message to the part of the brain that normally is telling you you've been touched on your finger. So it turns out by cranking this current high enough, you can actually feel like something just happened on your finger. So there's this idea of perception. So there's a stimulus, which is just whatever is out there. But perception is kind of the conscious perception. Conscious awareness of the stimulus. Right, so if I have a small enough current here, even though there is a electric current going across my finger, I might not notice anything. You keep making this larger and larger and larger until you finally, it's enough that it sends a critical threshold that you detect as something's happening at my finger. So that's where we get to this idea of a sensory threshold or perceptual threshold. This is like the minimal stimulus that is just noticed. Right? This could be, what is the quietest sound you can play on this thing that you just barely hear? Oh yeah, there's a sound. It's not silent anymore. Or what's just the lightest touch that you can feel where I actually feel it's a touch? Or what's the minimum amount of salt you can add into this where I know that it's salty and not just unsalted? So perceptual threshold is like, what's the minimum that I can have for the stimulus that I just barely notice? So that's going to be important. This is going to be our independent variable in all of these experiments. Not our dependent variable. Basically, we're going to have, here, let's put the finger the other way because it's easier. I'm going to take some electrodes. I'm going to put this actually, it's going to be a current that goes across there. And we'll talk about a little later why that's important. So this is some current source, which is going to send current through your skin. And our dependent variable is the perceptual threshold. That is the oh shoot. There we go. Minimum current that you just barely feel. Yeah, how are we going to measure this? How are you going to actually measure this? How are you going to actually measure this perceptual threshold?
Those electrodes have like a voltage. They say how much current, and if there is any stimulus, you say, ah, I felt it, and you take a measurement when you felt it.
Yeah, so you're going to start with something less than they can feel. Pick some value. This is going to be in milliamps. That's how you measure the current. You'll pick some value that is below what they can feel, and then increase it. Do you feel it now? Increase it more. Do you feel it now? Increase it even more. Do you feel it now? Increase it even more. Do you feel? At some point, they're going to like, whoa, I feel it. That is the value for this threshold. That's the minimum amount where they just barely felt it. Everything below that, they didn't feel. So in all our different cases, whatever condition we're doing, you are basically going to use the same process to get this threshold value. You're going to start with some value of the electric current that's below what is perceived, what's below what they're going to feel. And then you'll keep increasing it little by little. And at some point, they'll feel it. And when they feel it, that's the value. That is that minimum current that they can feel under that particular condition. So does that make sense?
Professor, one question. So the electrodes, the measurement would be the independent variable, sorry, each of the measurements?
We haven't talked about independent variables yet.
Okay, sorry.
So right now I'm just talking about the dependent variable. This is the thing that we're going to be looking at. We're looking at, here's the system where we're putting an electric current through your skin, finding what's the minimum current that you can just barely feel as this little tweak, as something happened. And then we're going to look at different conditions that might affect this perceptual threshold that would make it different. One thing we're going to do is body part. When we think about independent variables, one independent variable is going to be body part. We're going to do this on your finger. We're going to do this on the back of your calf. So again, let's look on the finger and see what the perceptual threshold is. Let's look on the back of the calf and see if the perceptual threshold is different. So, and this is independent variable 1. We're going to have 2 independent variables in this one. So what do you think? Does anybody have just any off the top of their head what you might expect trying to find the perceptual threshold on the finger versus the calf?
Your finger will have a threshold because there's more receptors on your finger, or they're more sensitive?
Exactly. You think, everybody's pretty, you know from your Bio 110 lab, your fingers are more sensitive to touch. You have more of these receptors here, which means that if you're running some current through there, there's much more of a chance you're going to have more things hitting some threshold and sending a message to your brain. If you have the calf and there's way fewer of these receptors under there, you're probably going to need more current going through to get sufficient signal going up to your brain to let you know something's going on. Right? So one thing we're going to do is that we'll have the independent variable be the body part. You can kind of think about it being spatial summation even. We're having more of these things all kind of getting hit at once that are all in kind of a close proximity to each other. We're, in the finger and less in the calf. The other independent variable is going to be the frequency of the stimulus. We use hertz, also cycles per second. Frequency is how many times per second something's happening. Like the things I was showing right now has just been a single stimulus. A single stimulus is you send a stimulus and you're done. You just stop. If I am 1 hertz, this is 1 cycle per second. That means you're going every second. You're sending off another one, like boom, 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 boom. If I was doing 2 hertz, I'd be going boom, 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 boom. You know, in fact, it's probably worth briefly.
All right, so this little piece of gear here is going to be our electrical stimulator.
All right, hold on.
It does lots of other things. We're going to be using it to measure your electrocardiogram and spirometry. But for today, we're going to use it purely run on this very simple repeat rate.
So I'm going to show, I hooked this thing up to my muscle so you can see this is not what you're doing. You're not hooking it to your muscle. You're doing it to sensory, but this gives you a sense of frequency.
You know, 20 milliamps is going to be enough to notice. And if you show my hand here, if I say send out a pulse, you know, you can see my thumb go. So I'm going to turn on the pulse. Here we go, pulse, my thumb went. Right now, the repeat rate is at 0.1, which actually means like 1 pulse every 10 seconds. So if it's set here, if you just turn it on, you'll have a pulse. As long as you turn it off before 10 seconds is off, that will be the same as doing a single pulse. There is no way to just tell us to do 1 single pulse, but you know, it's the same difference if we say 0.1 hertz and then make sure we turn it off before 10 seconds have passed. Now what I'm going to do is I'm going to change the frequency to 1 hertz. And you'll see it's sending out 1 pulse every second. So this is important to realize. 1 hertz is very different from a single pulse. 1 hertz means there's 1 pulse coming out every second as long as the stimulus is on. You know, a single pulse would be there's a pulse and then nothing. I can increase it. I'm going to go to 2 hertz, which is 2 cycles per second. And now all of a sudden my finger's going twice as fast. I'm going to go right now and change it to 5. You know, I can go even more. I can take it to 10. You know, at this point I'm starting to get into tetanus. It's actually getting kind of uncomfortable. So I'm going to like slow it down and turn it off. Ah.
All right, so the idea there is just kind of showing you.
So that gives you a sense.
So does that make sense? The difference between like, what hertz are? Like, so if you're running this thing at 1 hertz or 5 hertz, that's how many times it's coming out per second. What do you think that is going to do in terms of threshold?
It'll lower the threshold because of temporal summation.
Exactly. Right? If I have, in fact, let me stop sharing this because we're just kind of there. If I am sending out pulses in quick succession to the same little piece of skin, we're going to start adding up. The faster in succession they are, the more they're going to start adding together by this temporal summation. So I'm not going to need as high of a value of milliamps to actually hit my threshold. I can have a lower value coming out of the electrodes, but since they're in fast succession, the overall effect at the neuron in terms of bringing it up to thresholds going to be the same, and I'll feel it. So does that make sense? So when you're writing your hypotheses, make sure you think about, there's independent variable. There's two of them. One is there's body location, but the other one is the frequency of the stimulus. So in both cases, you're going to look at two versions of body location, various values of frequency of the stimulus. Both cases, you'll see how do those affect my dependent variable, which is its perceptual threshold. And again, this is going to be measured in milliamps. This is going to be what is the lowest current that I deliver that I just barely feel. And your hypothesis will have to have two parts to it. It's going to have to be what's body location versus perceptual threshold and why, and what is frequency versus perceptual threshold and why.
So for that first one, you could say, like, I think the finger will have a lower threshold because blank, and then compare it to the calf, or?
Correct, exactly. Exactly. So are there any other questions about this? I know we're like 15 minutes past, but this will all be uploaded, so anybody who had to leave will be able to see it still as well. Any comments, questions?
For this last part that you were just describing, so for example, a hypothesis could be that the higher the hertz, the lower the perceptual threshold would be because of temporal summation?
Yeah, exactly. Exactly.
Okay, thank you.
So, any other comments, questions? So I have this due like at the end of the weekend, so that gives me a chance to double check everybody's to make sure they all look good. But so try to get your pre-lab done by Sunday night, which that gives you like 4 days.